This is my audience. 1, 2, 3, 4, 5. And I have my laptop for my presentation. And then everybody else has that iPad and we'll be looking on. So, yeah. And Ellen's going to film. Tell me when you're ready.
Ready.
Okay, hi guys. So today I'm going to give you guys a presentation on the translation of DNA to protein. So first of all, have you ever wondered why do we have so many genes? Because we all have genes and they're all being expressed. And you ever wondered why exactly, how are they expressed? Why do we have them? So according to chem biology, genes are expressed as proteins, but how? And the way they're expressed is through RNA, which is our transition language that we use from DNA to proteins. So first we're going to start off with DNA, and if you look at the first slide, DNA is required for translation. It contains our information. It is what we use as our language or code. It is a really basic molecule. It's double-stranded. It's stable because the hydrogen bonds in between, they face each other, like the base pairing, so it's stable, so it's hard to lose information or for it to become mutated or damaged. So that's why we use DNA. There's 4 bases: guanine, cytosine, thymine, adenine. If you look at the PowerPoint, you can see the bases and you can see how they're bonded, and you can see that they're complementary to each other. So cytosine and guanine go together, and adenine and thymine go together. And so if you have one strand, you can replicate the other strand based on the opposite pairing. So gene expression is the process by which DNA directs synthesis of proteins. The information in the DNA needs to be translated to RNA. It needs to, the message needs to be carried and expressed. So the message is expressed in RNA. It's that in-between language, because the language of DNA and the language of proteins are different. So there are 2 stages in which gene expression occurs. We have transcription and translation. And if you look at the next slides, if you look at the second slide, you can see the process of transcription and RNA processing. So you can see overall what does it look like going through. You can see the pre-RNA and the mRNA. And if you look at the third slide, sorry guys. Okay, so as I said earlier, we have 4 bases and there are 20 amino acids that proteins use. So because the language is different, we need codes or anticodons, but we're going to say codes for now because the first step is using codes, that can be a conversion factor for the bases and amino acids. So an amino acid code uses 3 nucleotide bases. There are 4 bases, so you can make up to 64 codes altogether. But there's only 20 amino acids. So a lot of the codes code for the same amino acid, but not one amino acid will have multiple codes. And you can see on the third slide that chart, you can see at the top, you have uracil, which is a substitution for thymine and RNA, and the rest of the bases are the same. So you can use the chart to go across and down and know what 3 codons code for a specific amino acid. And so genetic instructions for a polypeptide chain are written in RNA, but the coding sequence can be lapsed or can become out of the reading frame. When you read 3 bases at a time, it's called your reading frame. And if there was an insertion or a deletion, like a mutation, the reading frame can be altered. And so you have to always make sure that your reading frame is current and not out of place, because then you'll end up with the wrong 3 amino acid code nucleotide bases, and it'll code for a wrong amino acid. But the central dogma that, according to molecular biology, that they use is that from DNA to RNA, that RNA is that language. And there are 3 types of conversion languages. You have mRNA, RNA, and then tRNA. And then for mRNA is your messenger RNA. It's your primary script, your primary transcription. For DNA, it's the direct you use a complementary pairing, and you have a coding template and a just regular template. The coding template has the same base pairing as the mRNA strand, because the mRNA strand is opposite of the template used to replicate. So because of the opposite pairing, template DNA and mRNA DNA will have opposite pairing, and then coding DNA and then mRNA will have the same pairing, like base pairing. So if you look at the next slide, you can see that the primary or pre-mRNA is when you have you have your expressed genes and your non-expressed genes. And the non-expressed genes are called introns, and when you code for protein, the inexpressed genes need to be cut out. So the mature copy of the mRNA is when you only have exons. Those are your expressed genes. So to get rid of the introns, what we do is you cut them out using an enzyme called spliceosome. Basically, it cuts the two ends of the introns out, and then the two ends of the exons become joined together. So you can see that in the picture there. You can also see, if you see on the bottom, that on the left side you see a 5', and on the right side you see a 3'. The 5' and the 3' are caps at the end of the expressed DNA. The 5' is a bunch of guanine bases, which you still see in DNA, and then the right side is a bunch of adenine nucleotide bases. So they're basically there just to protect the ends when it goes through the translation process, which is the second step of protein synthesis. So the second type of mRNA is, the second type of RNA is ribosomal RNA, which is called rRNA, and basically it's the RNA that is used to make up ribosomes. So it codes for ribosomes, because ribosomes are what you use, the enzymes you use to make proteins, but they are also made out of proteins. So it's a special type of RNA to make the machinery needed to make proteins. And then your third type of RNA is transfer RNA. And transfer RNA is called tRNA. And basically what transfer RNA does is it holds your amino acid, the corresponding amino acid, to the mRNA codon. So it's like you have an amino acid on top and you have an anticodon, like 3 prongs on the bottom, and they connect to the mature mRNA. So when they relate at the bottom to make sure that they have the correct amino acid attached to the top. And you can see in the picture that it's got a weird shape. It's an L shape. You can see the anticodon at the bottom, and you can see how you have the mRNA at the bottom with the codon. They have complementary base pairing. That's how you confirm that you have the right amino acid, that the tRNA is carrying the right amino acid. You can see a 3D model of the tRNA there. And then, so once you have the correct RNAs in place, you're ready to conduct protein synthesis. And so you have 2 steps, like I said earlier, transcription and translation. And so in transcription, you can see in the next slide, the top part, you can see in the nucleus, that's where transcription takes place. DNA, you have your template strand, and then your primary mRNA strand is made from the complementary base pairing. That is spliced, and you have your mature RNA, and that whole process is called transcription. So they're all overlapping processes. That whole section or process is called transcription. After you have your mature mRNA, it leaves the nucleus through nucleopores, and it's in the cytoplasm, which is where protein synthesis will take place. So now you're ready for translation, and there are 3. Translation is divided into 3 sections. You have initiation, elongation, and then your termination. What happens is the beginning of, you have your ribosomes that come together. So there's a large subunit and a small subunit, and you can see it in the picture. The bottom is small and the top is large. The tRNAs fit inside the top portion, the large part of the ribosomes, and they carry a amino acid. That amino acid is placed there through another enzyme called aminoacyl synthetase. It's a long name. Just know that the tRNA is attached with an amino acid by that other enzyme. But it comes in, fits in the ribosome, and then it finds the starting region on the beginning of the mRNA. So they attach the anticodon and then the codes of the mRNA, they attach together and just initiate to make sure that, like a confirmation signal sequence basically. And then it moves along, and the amino acids attach onto the next tRNA. You have three types. You have an exit site, an A site, and a P site. And so the P site attaches to the A site, and then the P site leaves. And so you keep having you keep having tRNAs that come in, and then they attach from one to the other, the amino acids, when the mRNAs on the bottom keep moving along, basically. So you'll have a long chain of amino acids, and then once it hits the stop region, then it just pulls off, and then the whole complex disassembles, and so you have a long primary structure of proteins. And then it'll spontaneously form, and you have a protein. But a couple mutations that you can have when this process happens is a point mutation and a silent mutation. A point mutation is when you just have one base that's changed, and it's just a substitution of bases. Sometimes it doesn't produce a huge effect, sometimes it does. A silent mutation is when it doesn't produce a huge effect and you don't see anything. Missense mutations is when the substitution occurs such that it doesn't really code for anything, and so it either codes like for nothing at all, or it codes for a stop codon in the middle of a protein, and when it tries to be expressed, it doesn't work, like it's just a dysfunctional protein. But so that's the expression of genes. So you can see that genes are a critical point in the expression of proteins to get proteins. Now we can all appreciate the features of genes, and